Hello, everyone. My name is Alexandra Santos. I'm a professor of Pediatric Allergy at King's College London and an attending physician in Pediatric Allergy at the Evelina London Children's Hospital. It is a great pleasure to join this Food Allergy Awareness Week organized by FAIR, and I would very much like to thank FAIR for the kind invitation to join this online event. And I will spend the next few minutes talking about new tests that are emerging, making the transition to clinical practice, and that can help us to improve the accuracy of food allergy diagnosis and also the comfort and making the process more comfortable and easier for patients and families. So it is extremely important to get the diagnosis of food allergy right. Food allergy has a huge impact on the lives of patients and families and society in general. And so we really need to be able to, for every patient and every food, be sure whether or not the patient is allergic. Because diagnosing food allergy will impose a lot of restrictions, dietary restrictions, but also social restrictions. It will have some psychosocial impact. So we need to be aware of that. And not really make a diagnosis of food allergy lightly and be very determined to get an accurate diagnosis. So currently, the cornerstone of the diagnosis of food allergy is the clinical history. So that time that the doctor takes to speak to the patient, to the family, and ask about symptoms, previous exposure to the food, whether it has been consumed or not, where there were symptoms suggestive of allergic reactions, whether there's other risk factors to make the for food allergy or for severe allergic reactions to foods, there's a family history of allergic disease and so on. So for some patients, there's a clear history of an immediate type allergic reaction to a food. For other patients, there is a clear tolerance with the food being part of the diet and being consumed very frequently without developing any symptoms. And there's also patients that have never eaten the food, or where the food is involved in a possible allergic reaction where it's not clear-cut which was the food implicated, if it was really an allergic reaction. So things sometimes can be a bit less clear from the history. The next step in the diagnostic process is to look for evidence of IgE antibodies against the specific food. So the IgE antibodies are the allergy antibodies. And when an immediate type allergic reaction to a food, including anaphylaxis, our body produces IgE antibodies against the specific food. And we can look for this using skin prick test or specific IgE in the blood using a blood test. If we can make a diagnosis based on the history and the results of these tests, then we don't need to do any more testing. If the results are equivocal and we are not sure whether or not the patient is allergic, the gold standard to diagnose food allergy is oral food challenge, which consists in bringing a patient to the hospital or to the physician's office and give small amounts of the food at set intervals in increasing amounts up to an age-appropriate portion of the food. And then monitoring for the development of allergic symptoms. At the first signs or symptoms of allergic reactions, the challenge is stopped and the symptoms are treated. And if the patient is able to consume the food up to the total dose of the food planned for the challenge, then the challenge is considered negative and the patient can incorporate the food in the diet. But try to reduce the number of challenges that we need to do, because obviously they involve the risk of an allergic reaction, and they involve a lot of logistics and resources and experienced team and resources to treat allergic reactions of any severity. So we try to do tests before we need to refer a patient for an oral food challenge. At the moment, the tests we have are the skin prick test and also specific IgE testing, which is a blood test where we look for the allergy antibodies in the blood, either to an allergen extract. So for example, hazelnut extract, which is a mixture of all the allergens and different components from the food source, We can also use what we call component resolved diagnostics, which is looking for the Ig antibodies to a specific protein within the food, so a molecular allergen. And we try to make the most of this information to try to reduce the number of patients that we have to put forward for challenges. And so this is a figure illustrating some of the common allergenic foods and then some of the individual allergens that exist as part of this allergen source. So when we say we are allergic to egg or cow's milk or peanut, actually different patients can mount an allergic response to different allergens within egg, within cow's milk, and within peanut. And then within this mixture of allergens that exist in the food source, some allergens are known to be more potent allergens, and others are known to not be able to cause significant allergic symptoms. And so identifying Ig antibodies to these specific allergens can be more informative than determining Ig to the whole extract for some foods. And the reason for this, one of the reasons for this is that, for example, patients with respiratory allergies and pollen allergy, they develop Ig to the pollen proteins. And then they can recognize that these Ig antibodies can recognize allergens in plant foods, such as peanut or peach, that are very similar to the pollen allergens. And these are so-called cross-reactive allergen components, so cross-reactive allergens. And these allergens in the food, in the plant foods, are usually labile proteins that are easily destroyed by cooking or by digestion. And so the symptoms are usually mild and are usually limited to the oral cavity, the throat, and sometimes in some patients, some abdominal pain. But systemic reactions are extremely rare. They are possible, but they are extremely rare. So identifying Ig to individual molecules helps us to identify, for example, within peanut, whether the Ig is directed to the real peanut allergens or is directed to the other cross-reactive allergens. And so it can help us sometimes to distinguish for some patients whether they have true peanut allergy or whether they have pollen food syndrome. So whether they have a positive test to peanut because of their pollen allergy rather than real peanut allergy. And so this is one of the examples where determining Ig antibodies against individual molecules can be more helpful than identifying the Ig antibodies that react to the whole allergen extract. And so because we know the primary pollen allergens, we know the primary plant food allergens, so the ones that are sort of able to cause systemic reactions. And then we know some of the cross-reactive allergens that are not usually able to cause systemic reactions, like, for example, pre-R10 proteins, profilins, so which are allergens involved in this phenomena of cross-reactivity. Lipid transfer proteins are other types of molecules that can cause systemic reactions in some geographical locations. But so the general message here is that determining the Ig antibodies to individual molecules can sometimes be more informative than determining Ig to the whole extracts. And so one of these examples foods is hazelnut. So within hazelnut, there are these allergens that are cross-reactive with birch pollen, like core A1. And then there are other allergens that are sort of primary hazelnut allergens like core A9 and core A14. And so here in these graphs, you can see the levels of Ig to the various allergens, and then the number of true positives, true negatives, false positives, and false negatives for Ig to each of these allergens. And then the proportion of patients that were correctly diagnosed or incorrectly diagnosed based on these different Ig to these different allergens using different cutoffs, so using different levels for positivity. And so generally what you can see is that determining Ig to core A1, which is the cross-reactive allergen, doesn't really, so reduces the number of so it doesn't really help us to get more correct diagnoses. And using a core A9 and core A14, which are the primary hazelnut allergens, can help us to increase the number of patients that were correctly diagnosed. And this is because we can essentially these are primary hazelnut allergens, and they do not take into account the other allergens that are involved in phenomena of cross-reactivity and can cause false positive results. Another example is peanut, because in peanut it's another plant food that has allergens that are cross-reactive with the tree pollen or cross-reactive with grass pollen. But of course, there's also primary peanut allergens like RH2 and RH6. So, which are the best to identify, real peanut allergy, as you can see here from these ROC curves. So these ROC curves are curves that relate the sensitivity of a test with 1 minus specificity. So the best test is the one that has the IG, so the curve closest to the upper left corner. And so the closest to the upper left corner, the more discriminatory the test is. The closer to this reference line here, the more it is sort of completely random, the result. And this is what happens with specific IG to RH9 in our population, or RH8, which is one of the cross-reactive allergens, and then RH2 and RH6, which are potent allergens and are really the ones that are the most useful to identify peanut allergy, are really, they really stand out compared to the other peanut allergens, or compared with peanut extract here in light blue. And so here on the right, so this is work that one of my PhD students did in the lab, comparing the importance of RH2 and RH6 in peanut allergic patients recruited from our pediatric allergy clinic in London. And what we saw was that 84% of peanut allergics in our center had RH2, IG to RH2 and RH6. But RH2 was able to cause the granulation of mast cells, or a reaction of mast cells, which are cells involved in allergic reactions with a lower concentration of the allergen and to a greater extent. So with a greater number, a greater proportion of mast cells that become activated. And RH2 was also able to inhibit, so suppress binding of IG to the other allergen RH6 more than RH6 was. So overall, we concluded that RH2 was the most important peanut allergen in our population in the UK. Now, another area where individual allergens can be more informative than the extracts is to identify children that are able to tolerate baked milk or baked egg whilst being allergic to milk and egg. And the reason for this is that there are some proteins within milk and within egg that are more susceptible to degradation by temperature. So they're more thermolabile. And therefore, they can be broken down when the food is baked. So when it's extensively heated for at least 20 minutes at least 180 degrees Celsius. So, and what happens, so these conformational epitopes, which are parts of the allergen that come together when the protein is sort of in its natural form, but then are broken down when the food is cooked, can be identified in some proteins, individual proteins within milk and egg. So, for example, so one of these proteins is casein, which is a thermoresistant protein. So it's more resistant to temperature than other proteins within cow's milk. And so IG to casein, when it's high, helps us to identify a patient that is less likely to tolerate baked milk. And similarly with baked egg, so ovomucoid is a protein that is thermoresistant more than ovalbumin, for example. And so it's determining IgE to ovomucoid can be more helpful than determining IgE to ovalbumin or to the whole egg white allergen extract to identify patients that are going to react to baked egg. Now we can go one level up. So we can use allergen extracts, as I mentioned, we can use individual allergens like ovomucoid for egg or casein for milk, but we can go one step up and actually look for Ig binding in specific parts of the allergen, specific peptides, so specific short segments of the molecules. And these can be even more informative in some cases. So we have looked in collaboration with Sahila Maleki and Barry Holbert from the US, we have looked at IgE to peptides of all peanut allergens at the time that we did this. Now there's a few more that have been described since, but we determined Ig binding to the whole sequence. So we broke it down all the allergens in small peptides, and then we determined Ig binding to these peptides. And so we then compare the peptides that were recognized by IgE of peanut allergic and by IgE of children that were peanut sensitized. So they had IgE, but they were not allergic. And then we also had some non-sensitized, non-allergic controls. And so we did extensive analysis trying to find which peptides were most like discriminatory between allergic and tolerant children. And we identified 7 peptides that were bound more by these IgE antibodies of allergic children compared to tolerant children. And so they all fell within Rh1, 2, and 3, which are the most important peanut allergens, particularly Rh2. So there were 2 on Rh1, 4 on Rh2, and 1 on Rh3. Interestingly, there was also a peptide that was recognized, a peptide on profilin, which is one of the allergens involved in this phenomena of cross-reactivity, that was bound more by IgE of tolerant children than IgE of allergic children. Which was interesting because, again, these cross-reactive allergens can lead to false positive results. And so this is the heat map showing where there's more Ig binding, it's more red. When there is less Ig binding, it's more blue. And in this horizontal line here of red, you can see the results for the allergic children, and in green, the results for the tolerant children. We then validated these findings using another method that is very commonly used in allergy clinics all around the world. And we collaborated with a company that sells these tests and asked them to bind our peptides to their platform so that we could measure Ig binding to these peptides using this commonly used method. And so with this new method, we could confirm our findings in the microarray, and then we could also try to see whether these peptides could be useful diagnostically. And so we started by comparing the levels of IgE to these peptides between allergic and sensitized but tolerant children that are identified here as PS. So PA is allergic, PS is sensitized but tolerant children, and then non-allergic controls. And we looked also in parallel to Ig to the whole allergen. And essentially what we could see was that only the peptides, 3 peptides from Rh2 and 1 peptide from Rh3 were actually significantly different, the Ig binding between allergic and sensitized but tolerant children. And when we looked at the diagnostic utility of these peptides, only the Rh2 peptides actually proved to be useful. And we found that taking the 4 peptides that we had identified all together and together with Rh2 specific Ig allowed to improve the diagnostic performance of Rh2 specific Ig, which is already very good. So we could increase the positive predictive value and the specificity of Rh2 by adding these peptides by 7.7%. Which is useful because it can help us to confirm allergy in some patients where we wouldn't be completely sure just by measuring Rh2. Now there's a new method to measure IgE to peptides using a bead-based array. So where peptides, instead of being bound on a sort of platform, a microarray, they are bound to beads, and then it's the same principles. Then we add plasma serum that then binds of the patient, then Ig will bind to these peptides, and then we are able to measure this binding with another antibody which is conjugated with the fluorochrome, so emits light, and then we can measure by the intensity of this light, we can know how many antibodies there were bound to the peptides initially. And so this bead based array has been, so bead-based epitope array has been developed for peanut allergy, and its diagnostic utility has been studied in the LEAP study, and it has been validated in 2 independent cohorts, the COFAR cohort and the POISED cohort. And so the sensitivity and specificity of these tests was 93 and 94% respectively, with a positive predictive value of 91% and a negative predictive value of 95%. So we can look at also other characteristics, other functional characteristics of IgE, of the IgE antibodies. So we can look at specificity, which is what we've just talked about. So the epitopes, the parts of the allergen that IgE recognizes. We can talk about avidity or affinity, which is the strength with which the IgE binds to the allergen. We can talk about specific activity, which is the proportion of the whole IgE of the individual that is able to recognize the allergen. And then diversity, which has to do with the different allergens within a certain food source that the IgE can recognize. And this is again work from Ollie Hemmings, one of my PhD students, who showed that the greater the levels of IgE, the more activation of the cells involved in allergic reactions we observe. The stronger the reaction between the IgE and the allergen, the more mast cells will respond, mast cells and basophils, which are the cells involved in allergic reactions. The greater the proportion of IgE that is allergen specific, so that is directed to that specific allergen, the more activation of mast cells and basophils we see. And then the more diverse response of the IgE antibodies, so the greater the number of allergens within the food source that the IgE recognizes, the greater the activation and reaction of mast cells and basophils in vitro. So this is a summary of the work from Ollie Hemmings that I just described. And so he was able to see that all these parameters of IgE contribute to the amount, to the number of mast cells and basophils that are going to react to the allergen. And we have related this number to the likelihood of having an allergic reaction and also with severity of allergic reactions. And so he saw that it's the diversity of the IgE response and the proportion of IgE that's allergen specific were the greatest determinants of basophil and mast cell activation, although all these factors contributed independently. So he also tried to see whether we could use this information to improve the diagnosis of peanut allergy. And in fact, if we consider the functional characteristics of IgE, so the sort of quality of the IgE, in addition to the levels, we can improve the diagnostic accuracy of the levels of IgE. So he built these diagnostic models showing a sensitivity of 87% and a specificity of 85%, with a positive predictive value of 89% and a negative predictive value of 82%, and an overall diagnostic accuracy of 86%. So this is better than peanut-specific IgE, but it's not better than other tests that we have been working on. And you'll see that in the next few slides. So the other promising next generation of food allergy diagnostics are these cellular tests, which look at the response of the cells involved in food allergic reactions, such as mast cells and basophils in vitro, as opposed to actually exposing the patient to the allergen. We look at the cells responsible for allergic reactions in the test tube. And so this is called the basophil activation test and the mast cell activation test. So the basophil activation test, we use blood from the patient, a very small amount of blood from the patient. We add the allergen or controls, and then we add antibodies that allow us to stain for specific markers to isolate the basophil population. And then within this the basophil population, by flow cytometry, we are able to see whether the cells are activated, so whether they reacted, whether they degranulated or not, in response to allergen and in response to control. So we have negative and positive controls. So they always have to respond to the positive controls. And then we can see whether they respond to the allergen or not. In the mast cell activation test, it's slightly different. So we use cells that are grown in the lab, human mast cells, but are a cell line. So they are immortalized, they're the same. They are all homogeneous, so they are all the same within in the flask that we are growing in the lab. And then we add plasma or serum from patients. So this is the part of the blood without the cells. So and that contain the antibodies. The antibodies will bind to the receptors of the mast cells. And then it's like mimicking the patient's own mast cells. And then again, we add stimulants, so allergen or controls. And we can measure the activation by flow cytometry, similar to what we do with the basophil activation test. And so we have done a large study a few years ago assessing the utility of the basophil activation test to diagnose peanut allergy in the UK. And we saw, as you can see in this graph, that the basophils of peanut allergic children had a high response and a dose-dependent increase in activation markers with increasing concentrations of peanut extract. And the basophils of children that had IG but were not allergic did not significantly respond to peanut. We identified an optimal cutoff, and we were able to see the performance of this test. And overall, the diagnostic accuracy was 97%. We were also able to see how this test performed in a new population. And here specificity and positive predictive value reached 100%. So indicating that this test is very useful to confirm the presence of peanut allergy. We've had the opportunity to apply exactly the same method to participants in the LEAP study, which was a very big population of patients, very well characterized. And we applied exactly the same method, and we applied this same cutoff, which is the way we interpret the test as positive or negative. And we could see a very high specificity and positive predictive value, confirming the utility of the test and its sustained very good performance and the utility particularly to confirm the diagnosis of peanut allergy. We tested samples from the same cohort of patients that were recruited from clinic to be assessed for possible peanut allergy on the mast cell activation test. And we could see that the cells that had plasma from peanut allergic children were able to respond to peanut in the vast majority, whereas the plasma from children that had IgE but were not allergic were not significantly responsive. However, their response to the positive controls was very, very similar. But as you can see here, so that we have IgE levels on the x-axis and mast cell activation on the y-axis. So for children with the same IgE levels, we had different levels of mast cell activation. So this is where it's really useful. We have also seen that patients with severe or life-threatening allergic symptoms during food challenges had high levels of basophil activation and high levels of mast cell activation, a proportion of mast cell activation, which can help us to identify the patients that are more at risk of having an adverse event during food challenges. So we've generated models from this data, and we have put this together in an online calculator that can be used by healthcare professionals to compute, based on the tests alone, what is the probability given by the tests of a patient reacting during an oral food challenge and of having a severe reaction during an oral food challenge. So it does not it's not an overall clinical assessment, it's just computing based on the results of the tests and based on the data from this study, what is the probability of having a reaction, having a moderate reaction, or having a severe reaction. So essentially, we could try to do these tests that we currently have, so skin prick tests and specific IgE to diagnose patients as much as we can. But for the patients that would need to be referred for a challenge, we could do these additional tests. So the epitope testing, IgE to the peptides, or basophil and mast cell activation. And so if the tests are positive, we can confirm the diagnosis of food allergy and recommend allergen avoidance. If the tests are not positive, then we would do an oral food challenge. But because there's still a proportion of patients that may react with these negative tests. So with negative basophil activation tests or with negative IgE to peptides. And so, but the chances that these challenges will be positive would be much lower than currently, because a lot of the patients that were allergic were excluded by doing these tests before referring them for a food challenge. And we have confirmed the effect of this approach in reducing the number of oral food challenges in the peanut study that I just mentioned, and also in the pronut study, which looked at peanut and tree nut allergies, and also a reduction, but this was particularly a reduction in the number of positive challenges, which is really what's desirable, because these would be patients that would otherwise react during the food challenges. We are currently finalizing the development and validation of the basophil activation test and the mast cell activation test to other food allergens, including cow's milk, eggs, sesame, and cashew nut. And I'm very excited to start very soon a study funded by FAIR, which is a multi-center study across the UK with 10 centers in England and 1 center in Scotland, where children aged from 6 months to 15 years with suspected food allergy that require an oral food challenge for clinical reasons will be invited to participate in the study. And if they agree to participate, they will have a clinical assessment, skin prick testing, blood collection for specific IgE, basophil activation test, mast cell activation test. And then we'll also do some questionnaires, and children will be randomized to either have the standard of care, which is to have a food challenge, or take the basophil activation test and the mast cell activation test into account to decide whether or not they need to have a food challenge. And then they only have a food challenge if the basophil activation test is not positive. And so, and then the primary outcome of the study is the proportion of allergic reaction, so the proportion of children undergoing challenges that had a reaction during the challenges. But we'll also be looking at the effect of this approach in the number of oral food challenges, in quality of life, anxiety, and healthcare costs. So I'm really excited to start this soon. I would like to thank FAIR for the opportunity and the funding to undertake this very exciting study. And finally, I would like to thank my colleagues, so my group in the lab and in the clinical trials unit, and my colleagues in the clinic, and my collaborators, and of course my funders, the Medical Research Council in the UK, the ITN and the NIAID and FAIR in the US, and also the European Commission and ASMA UK and the Wellcome Trust. Thank you very much for listening.